Thanks so much for having us and giving us the opportunity to present our new tool, MethPhaser. So obviously I'm not talking about structure variants today. Of all, long reads are really useful for them, as you have probably heard already a couple of times today and probably tomorrow as well. But what we are focusing today on is really the phasing of it, and why do we need phasing, and how can we improve phasing with the utility or by utilizing methylation. And so long reads have been really instrumental of improving phasing over the years, not just assembly and structure variant detection. But before we dive into this a little bit more, I just want to give you guys all an example why phasing actually matters. We all agree larger phasing is better for one or the other reason. But one of the cool examples that I always go back to is this gene DPMT. So DPMT encodes an enzyme that metabolizes teobrine drugs. And so basically there are two variants known on this gene that you have to report if you see them in patients. The challenging here is that both variants can co-occur and they are around 8 kb apart. So now if those variants are in cis, meaning on the same haplotype, the patient has still a functional gene and can metabolize the drug that the doctor might give him. If the variants are now in trans, the patient doesn't have a functional gene, and basically the doctor can kill him by giving him drugs for his treatment, right? This is kind of a rare case, so don't panic. But this is a clear case where phasing really matters, that we understand how variants are co-occurring on the individual molecules. And so what are the techniques that we are using these days? Well, there are basically 3 of them. The first one is population-based phasing. This has been used a lot of times, where we do usually that on common variants, right? There are several limitations with it, because we are just using common variants. We cannot phase or give a lot of information about rare variants that we so often care in disease backgrounds, right? Also, it cannot phase novel variants, because again, we are just focusing on common variants. A little bit better than that is trio-based phasing, but then we not just sequence a population of human genomes, but we need to sequence the parents and the proband. The advantage here is we can also phase rare variants, but we still cannot phase de novo variants, which are often very important for Mendelian diseases, for example, right? So we are still lacking, actually, the thing that we want to have. And so here is where long-read sequencing comes into play. Long-read sequencing really enables us to not just phase common and rare variants, but we can also assign the novel variants if they are coming from the maternal or the paternal haplotype, which is really instrumental and really cool. However, not so fast. There's always a but in my presentations. There's a little bit of problems here. So on the top picture here, let me get this pointer thingy here. On the top picture here, you see how read length impacts the phasing ability. That's taken from a review paper that we published a few years ago. But also when you look at samples, so here's an IGV screenshot with haplotype 1 assigned, haplotype 2 assigned reads, we often get a lot of untagged, unassembled reads, which are then making it hard to connect 2 different phase blocks. And that's basically a function of your read length, right? So these ultra-long nanopores are very helpful for that, but they're still causing some issues in certain regions, but also, of course, the rate of homozygosity in these regions. We always can only phase heterozygous variants or heterozygous SNPs per se. And so, given these limitations, we were really wondering if we can utilize methylation signal on its own to overcome this kind of stretches of homozygous variants. And the short answer is kind of yes. So here you see already some results from our tool. You see in blue the SNP phase block N50 that we take as an input, for example, from WhatsApp or other phasing methods. And then in this orange color, you see our results. And you see on the y-axis the N50, so length-based and phasing results, right? On the x-axis, you see different samples. It's all HT002, but some of them are R10s, some of them are 9s, because for us it was important that this method really just works and not just on one particular flow cell model, right? And also at different coverage, so 30x, 60x, as well as 80x. You can see that the coverage per se is not that much impacting, but the initial ability of phase SNPs based on the read length is really the driving factor. And so the way how we are doing this is basically, I'm getting pretty big echo here, is basically the work of Eli Fu, who is a student who actually implemented MethPhaser, but unfortunately because of visa issues he couldn't be here, and that's why you guys have to listen to me. What we are doing is kind of an iterative step. So we have the SNP-based phase blocks, as dictated here by haplotype 1 and haplotype 2 on the left and the right. And then we iteratively assign, based on the methylation information, reads to these phase blocks, where the methylation information in the haplotype in the phase blocks corresponds to the methylation information outside. And so by doing this iteratively, we cannot just assign untagged or unphased reads to the individual haplotypes, but we can actually start closing these gaps iteratively and very straightforward. So then, of course, it becomes very cool because by closing these gaps, we can extend the phase blocks and combine the phase blocks very well, right? The trick now is, of course, where we have to be careful, and that's coming next in the evaluation, is that we don't introduce many more errors by doing this, right? Because there's a good likelihood that we are introducing switch errors where different phases of haplotypes are stitched together wrongly, or flip errors where single nucleotides are assigned to the wrong phase block. So this is important. So to look at this, we did this evaluation based on WhatsApp tools and compared to the benchmark of genome on a bottle. And so on the top, you see the same slide that I showed you before, the N50 increasement, and on the bottom, you see the error differences. And so what you see here is the error in blue of the SNP-based phasing, and next to it, the methylation-based phasing. And basically, they are identical. So most of the errors that are happening are actually little flip errors here, which were quite interesting for me to see, which means, again, that few single SNPs are assigned to the wrong haplotype by doing this method. And so overall, I think, as far as I remember, the switch error is just impacted by 0.2% or 0.02%. And so this really gave us confidence that this method actually works. Now, every biologist in the room will say, well, wait a minute, what about sex chromosomes? And you are absolutely right, it doesn't work on sex chromosomes, because there the association of the methylation is very random across the cells, right, to the haplotypes. That's also something I had to refresh my genetic lessons from college. So the next question is always, for us at Baylor, or in general, does it actually really matter? Are we just phasing regions that we don't care about, or is it really something really important? And so for this, we assessed it twofold. So first of all, we looked into challenging medical relevant genes. And there you can see on the table here, we can really reduce the number of required phase blocks to kind of phase these challenging medical relevant genes together. These are the 380 genes that we published together with Genome on a Bottle, and really kind of really bad genes, but really important, like LPA and other genes that we are caring about. On the right side, maybe a little bit cooler, is actually HLA, or two genes in HLA. I believe it was HLA-E and F, but I cannot read it anymore. The cool thing, so on top you see here, is with SNP-based phasing, again, haplotype 1 in red, haplotype 2 in blue, and then unphased reads below. And then with MethPhase, we get the reads assigned and we get the gap filled, so we can phase these two genes together in the sample. And the cool thing is we also kind of assign these unphased reads in these phase block regions, which also helps you to identify variants, haplotype-specific variants in these regions. And the third cool thing, of what we didn't systematically benchmark this, is with this we were also able to assign haplotypes to structure variants while doing this phasing on the SNPs. And this is really something cool, because everyone that looked into phasing of structure variants probably has seen that WhatsApp and other tools do a lot of errors in assigning haplotypes across SV regions. The last thing I was really, really curious to see, though, and that also pertains a little bit to one or two of the talks that we just heard, is does it work in other samples? Well, the short answer is yes. That's not a biggie. Like a couple of other ethnicities from the HBRC cell lines. That's pretty boring, right? But the really intriguing thing I wanted to benchmark is patient samples on blood, because like blood, you could have heterogeneous methylation, right, that can throw the program off. And plus, most of the time, the cell line data looks a little bit better than the blood data from patients in terms of DNA length and everything, right? So for the patient samples, we still see an improvement. You can see we are kind of can resolve or can join around 2,000 to 3,000 phase blocks on the whole genome, on the whole autosomes. The advantage here is not that huge because the blood samples had a pretty low N50, so the initial phasing of the SNPs were reduced, but we could still improve it. So one of the limitations here is very clear. First of all, sex chromosomes don't work, and second of all, we need a certain good length, read length, that usually happens with a good SNP phasing initially, and then the program really can utilizes the methylation information to improve these phase blocks further. So with that, just some conclusions. It's really the first method, as far as we are aware, that utilizes really the methylation signal itself. We can show that we're improving the N50 in 1.5 to 3-fold on different samples. We did test that across different ethnicities. We did also test this on blood samples, which I didn't walk you guys through very much, but I think this is really important that we also tested this component. MethPhaser is freely available on Todd's GitHub page. And so, like, yeah, the remaining is just thanking again Eli Few, who drove the project, and then together with Sergey from Oxford Nanopore, he started this in his summer internship in Nanopore. So, like, really great opportunity for anyone else who wants to check this out. And then Eli is a student from Todd that I'm co-supervising, and then my postdoc Matt had also contributed for the blood samples for the analysis. Thank you so much.